Good evening. Many thanks to Dr. Lears and the other meeting organizers for organizing this special symposium, for inviting me to present our work here. For the next 20 minutes, I'm going to tell you about the protein Junctophilin-2 in cardiac EC coupling and its novel function in cardiac transcription regulation we recently discovered. As already mentioned in previous presentations, it is well accepted that Junctophilin-2 is so far the most critical player in organizing cardiac or diet structure in cardiomyocytes. Junctophilin-2 couples T-tubulars and junction SR together with a very narrow space, allowing precise local control regulation of calcium-induced calcium release in cardiomyocytes. Here shows a schematic diagram of JP2 structure and functional domains. On the N-terminal, Junctophilin-2 contains 8 MON motifs separated by a joining region. These MON motifs have spatial affinity for plasma membrane, is responsible for Junctophilin-2 protein binding with the T-tubular membrane. On the C-terminals, the transmembrane domain spans across the SR membrane. In between them, Junctophilin-2 contains an alpha helix domain, which is thought to determine the distance of the junctional coupling. But our recent work shows that this alpha helix domain encodes a lower stress adaptive transcription regulator. Anyway, the main function of Junctophilin-2 is to tether T-tubulars to the junctional SR and keeps the two membrane system at a certain distance between each other during active EC coupling cycles. In addition to the primary role of Junctophilin-2 in the formation of cardiac dyes, a series of work from our lab and some other groups have also strongly supported the notion that Junctophilin-2 is required for stabilizing T-tubular network during EC coupling, especially under stress conditions. By tethering T-tubulars with junctional SR, Junctophilin-2 helps maintaining T-tubulars in an organized network. Unfortunately, this important EC coupling structure protein is downregulated in disease hearts. Here is a summary of publications that report Junctophilin-2 downregulation in different heart disease models, including human cardiomyopathies of different etiologies. These red highlighted papers are from our own group. We now know that in the event of Junctophilin-2 downregulation, cardiomyocytes will lose ultrastructure integrity, including T-tubular remodeling and loss of cardiac dyes, and therefore develop EC uncoupling and calcium handling dysfunction. Because under this setting, calcium release from renin receptor will lose the local control by Arab calcium channel, which are normally just 12 to 15 nanometer away. In the course of searching for the mechanism of Junctophilin-2 downregulation, we found, in addition to the post-transcriptional mechanism discovered by Dr. Shiqiang Wang group, there is also a post-translational mechanism critically involved in Junctophilin-2 downregulation. During heart disease, stress-induced elevation of calcium, intracellular calcium, lead to activation of calpain, resulting into calpain-dependent Junctophilin-2 proteinalysis and therefore degradation of Junctophilin-2. Here shows a simple example supporting the involvement of calpain in Junctophilin-2 degradation. In a mouse model of ischemia reperfusion, we found 30 minutes ischemia and 60 minutes reperfusion can induce Junctophilin-2 degradation accurately, which can be prevented by pretreatment of calpain inhibitor MDR28170. More specifically, using a in vitro calpain cleavage assay by mixing human heart lysates together with the human calpain-1 and calcium in a reaction medium, we found calpain can induce a time and calcium-dependent degradation of JP2, which can be blocked by EDTA calcium chelator and partially inhibited by carpane inhibitor. This is also true to mouse origin of Junctophilin-2. Together, this data suggests that Junctophilin-2 is a direct substrate of carpane. Then we wonder where carpane cleaves Junctophilin-2. Using online computer algorithm, we identified 3 potential cleavage sites, 2 on the N-terminal side and 1 on the C-terminals. Mutagenesis analysis in combination with in vitro carpane cleavage assay lead us to conclude that the C-terminal site is the primary, most sensitive one to carpane cleavage, because only deletion mutant around this C-terminal site can preserve the full-length JP2 and prevent the production of JP2 truncates corresponding to this cleavage size. We named the N-terminal product corresponding to this cleavage site JP2NT and the C-terminal one as JP2CT. Surprisingly, when we express EGFE-tagged JP2 or JP2 truncates in HGK cells, we found the N-terminal product JP2NT, but not the full-length JP2 or JP2CT, are highly enriched in the nuclei. To understand the molecular mechanism of JP2NT nuclear translocation, we performed in silico computer analysis. Surprisingly, we found a nuclear localization sequence on JP2NT, which is evolutionarily conserved among many species. Importantly, deletion of this sequence from JP2NT abolished the nuclear localization of JP2NT in HGK cells. This is also true in cardiomyocytes. In adult cardiomyocytes transfected with adenovirus carrying JP2NT, we found JP2NT was predominantly expressed in nuclei. And the deletion of this nuclear localization sequence, NLS, abolished the nuclear localization of JP2NT in the nuclei, suggesting that this nuclear localization sequence is required for JP2NT nuclear localization. Further in silico analysis, identified an alanine-rich region in the alpha helix domain from alanine 367 to alanine 402, and this alanine-rich region is highly conserved among many species from mouse, human, rat, ferret to zebrafish. And more importantly, this alanine-rich region shows characteristic of helix-turn-helix structure. The molecular biology people knows that helix-turn-helix structure is a major structure motif capable of binding DNA in all DNA binding proteins that regulate gene expression. So this analysis suggests that JP2NT may be a lower regulator of gene transcription. Interestingly, when we deleted this alanine-rich region from JP2NT, we found this JP2NT was completely separated from DNA, which was labeled by topo3. So you can see that the DNA and the JP2NT, as marked by the green, are completely separated, with this sharp contrast to the white-type JP2NT. So this data suggests that the alanine-rich region is a DNA binding motif and is required for JP2NT binding to DNA. To systematically study the genomic targets of JP2NT, we generate transgenic mice with cardiac-specific overexpression of HA-tagged JP2NT. Immunostaining confirms that JP2NT is predominantly localized in the nuclei of cardiomyocytes from transgenic mice. And the chip-seq analysis using the transgenic heart and the anti-HA antibody revealed that the JP2NT is concentrated in the gene-enriched region, and especially in the promoter and 5' UTR regions, as shown here. And further analysis shows that JP2NT binding is enriched at transcription start size. We may know that title box is an important component of gene promoters, which is responsible for transcription initiation in title box containing genes. To study whether Junctophilin-2 can directly interact with title box, we performed gear shift assay. Indeed, in vitro gear shift assay using purified recombinant JP2NT revealed that the JP2NT directly binds to title box. Here in this slide, you can see that the GST fused JP2NT can bind and shift the mobility of DNA oligos containing the wild type title box, but not the mutant title box. Again, gear-shift assay with a purified peptide containing only the aniline-rich region shows that this aniline-rich region specifically binds to the consensus of the title box, but not the mutant title box. Also, deletion of the null motif doesn't affect JP2ND binding with the title box at all. All this data suggests that this aniline-rich region is responsible for JP2NT interaction with title box. Using the same approach, we found this aniline-rich region can also directly bind or interact with the MEF2 response elements. We know that MEF2 is an important transcription factor regulating hypertrophy response in the stressed heart. We then ask, what does the JP2NT really do in the heart? To understand whether JP2NT directly regulates gene transcription, the first thing we did is microanalysis using samples from cultured cardiomyocytes infected with JP2NT or fallen JP2 or empty virus. Surprisingly, we found almost 2,000 genes were significantly repressed, and over 500 genes were upregulated in JP2NT treated cardiomyocytes. In contrast, the fallen JP2NT has minimal impact on gene expression. The right panels are mRNA plots of microarray data. The red dots are significant older genes, so you can see that JP2NT, but not the fallen JP2, has a big impact on gene transcription. Intuitive pathway analysis revealed that these significant older genes are related to many important signaling pathways, such as the insulin receptor, integrin, ERK, MAPK, NF-kappa B, etc., suggesting JP2NT may negatively regulate cardiac pathology remodeling by preventing hypertrophy, fibrosis, inflammation, etc. So the next question is, does JP2NT really do anything good in the heart in vivo? Interestingly, using the transgenic mice overexpressing exogenous JP2NT, we found JP2NT has no any impact on mouse heart at baseline conditions. But when we subject the mice to pressure overload stress, we found the transgenic mice are protected from developing cardiac hypertrophy and myocardial dysfunction, which have better ejection fraction and smaller heart compared to control mice. At the molecular level, we found JP2NT exerts significant impact on transcription modeling in response to cardiac stress. Here shows RNA-seq data from wild type and JP2NT overexpressed heart. These red dots indicate significant older genes following pressure overload stress as compared to sham operation. We can see that pressure overload induced a marked change in the transcriptome of wild-type hearts, and that response was severely attenuated in JP2NT overexpressing heart, suggesting that JP2NT plays a substantial role in cardiac stress response by suppressing transcriptional reprogramming. Then we wonder what would happen if we delete the nuclear localization sequence from JP2 genes. To answer this question, we made an NS deletion mutant mice using CRISPR-Cas9 technique. We confirmed the NS deletion abolished the nuclear accumulation of JP2NT following cardiac stress. Importantly, we found that NS deletion mutant mice are more vulnerable to cardiac stress, so that pressure overload induced bigger heart and worse heart function in the NS deletion mutant mice, while the animal is completely normal at baseline. So these results further support the notion that JP2NT functions as an endogenous cardiac protector against pathological challenges. In summary, I hope I have convinced you that the Junctophilin-2 as a structural protein is essential for cardiac EC coupling function. Our data also suggests that JP2 encodes a lower stress adaptive transcription regulator, whose N-terminal cleavage product serves as a protective mechanism antagonizing pathological cardiac remodeling by repressing stress-induced transcriptional reprogramming. Interestingly, a recent study from Junctophilin group reported that, different from Capen-1, Capen-2 cleave JP2 at a lower size, that is, the glycine 482 and threonine 483. And in this paper, they reported that the C-terminal cleavage peptide produced from this lower size by Capen-2 goes to the nucleus and promotes cardiac remodeling. More surprisingly, they showed that the C-terminal cleavage product from this novel cleavage size also has a molecular weight of about 25 kD, which is similar to the molecular weight of the C-terminal cleavage product as we shown produced by Capen-1 through the cleavage side, the cleavage at the site arginine 565, threonine 566. But we know that these two cleavage sites is 83 amino acids apart. How come these two cleavage products have the same molecular weight? This is impossible. So you may think that this could be due to a difference in experimental condition of Western blot experiments by two different groups. So the best way is to address this question and the discrepancy is to perform a side-by-side experiment in the same condition. We did compare side-by-side whether Capen-1 and or Capen-2 can cleavage JP2 at the same size. We created internal JP2 deletions overlapping this cleavage size and expressed the plasmid together with the Capen-1 or Capen-2 in HEK293 cells. Then we use protein lysis to perform in vitro Capen cleavage assay. As shown here in the left, Capen-1 together with the casein induced a JP2 cleavage, producing the 75 kD JP2NT and 25 kD JP2CT. And Capen inhibitor partially inhibited the production of the cleavage of JP2 by Capen. Deletion of the variance cleavage size doesn't influence Capen-1 mediated cleavage of JP2 at all. However, deletion of our cleavage size overlapping the arginine 565, 566, threonine 566 completely abolished Capen-1 mediated cleavage of JP2 and the production of the 75 kD and the 25 kD truncates. This is also true to the Capen-2. You see that Capen-2 and casein induced JP2 wild-type JP2 cleavage. the deletion of the various cleavage size does not influence the campin-2 mediated mutant JP2 cleavage, but cleavage deletion of the our cleavage size completely abolished campin-2 and calcium-induced mutant JP2 cleavage. So all this data together suggests that both campin-1 and campin-2 cleave junctophilin-2 at the same size at the arginine 565, threonine 566. To better compare the efficacy of campin-1 and campin-2 on the cleavage of JP2, we assessed the activity of each exposed to a range of calcium concentration using in vitro campin cleavage assay. The formation of the 75 kD JP2-NT and 25 kD JP2-CT by both campaigns were dose-dependently increased by calcium. However, the cleavage efficacy of campin-1 on JP2 is much more robust and more sensitive to calcium than campin-2. This data suggests that the efficacy of campin-2 cleavage on JP2 is much lower than that for campin-1. So in summary, I can confidently tell you that campin-2 cleave junctophilin-2 at the same functional size, the arginine 565, threonine 566, as does campin-1 instead of the glycine 482 and threonine 483 size. Campin-1 has a higher calcium sensitivity and cleavage efficacy on JP2 than campin-2. Lastly, but not the least, I would like to give my special thanks to my fellows, colleagues, collaborators, and former mentors. Without their excellent work and continuous generous support, I wouldn't have this wonderful data to share with you here tonight. Thank you for your attention.